So our lecture today is going to be focusing mainly on the digestive system diagrams, which are going to be important for the upcoming exam. Which consists mainly of the anatomy of the digestive system. For the full discussion of the digestive system, I will be posting a three session. Those are three small sessions covering the digestive system. They will include the diagrams, but mainly today in our discussion, I will be covering the diagrams. In the second half of this meeting, we're going to be discussing the metabolism, which is going to be our last chapter for this semester. And we're going to mention a little about the metabolism lab handout. Some of you did have questions about how do we complete the handout and how we're going to be submitting the handout. All right, so if you have any questions concerning the metabolism lab handout, we can answer this towards the end of the meeting. Any general questions before we start today, like concerning the blood and respiratory lab exam? There was a mistake in the lab exam. So an answer for one cell was replaced by another cell. So this has been resolved. So I did correct this on the exam. And for those who did miss a point for getting them wrong because of this mistake, I did add those points for you. So I did manually enter correct your grade. All right. For those who did have a concern about misplaced answer for the cells on the lab exam for the blood and respiratory system, this problem has been resolved. So for those who didn't complete or for those who did complete the exam yesterday, they wouldn't have this problem. For any person who did complete the exam before yesterday, I did add those points for you manually. All right, so first 5 stars diagrams that we're looking at today is going to be the diagram showing the different layers of the gastrointestinal tube. So those are the 4 main layers forming any part of the gastric tube. So we have 4 main layers. First layer to the inside is called the mucosa. This is my mucosa lining. Comes after that, underneath the mucosa, we've got the submucosa. And then we've got the muscle layer of the gastrointestinal tract tube. This is going to be the muscularis externa. And to the outside of the muscle layer, we've got the serosa. So again, we have 4 main layers building up any part of the gastrointestinal tract tube. First one is going to be on the inside. This is my mucosa. Underneath the mucosa, we've got the submucosa. To the outside of the submucosa, we've got the muscle layer, which is formed of smooth muscles. This is going to be my muscularis externa, or in some textbooks, they will call it musculosa. So if you see musculosa, or this is the same as muscularis externa, this is the muscle layer of the gastrointestinal tract tube. To the outside of the musculosa or the muscularis externa, we've got a layer called the serosa. If we're looking closely to the structure of the mucosa, if you see on here, we've got the epithelium. And the epithelium, if you remember, the type of epithelium located in your gastrointestinal tract, this was simple columnar epithelium, which is non-ciliated. This is the type of epitheliums that I have in my mucosa in most of the gastrointestinal tract tube. Remember, exceptions for this general rule is going to be the oral cavity, the pharynx, and the esophagus. Those three parts are not going to be lined with simple columnar epithelium. They are lined with non-keratinized stratified squamous epithelium. And remember, those are sites that are directly exposed to the outside, so I need a higher level of protection. That's why I have multiple layers forming my epithelial covering. Remember, under any epithelium, we've got connective tissue to support it. And the connective tissue in the mucosa is what we call here, this is the lamina propria. Lamina propria is this connective tissue layer that will be located underneath my epithelium in the mucosa. This one on here. So another layer of my mucosa is going to be the lamina propria, and this is simply an areolar connective tissue that would be supporting my epithelial line. To the outside of the lamina propria, I will see a very thin muscle layer. This thin muscle layer that you have, this is going to be the muscle layer of the mucosa, so we're going to call it the muscularis mucosa. Those are the three layers that will be building up the mucosa. To the outside of the mucosa, we've got the submucosa, which again is going to be formed of connective tissue similar to the ones that we have in the lamina propria. This is where you've got the blood vessels, lymphatic vessels that will be conducting everything that you're going to be absorbing here. To the outside of the submucosa, we've got the muscularis externa. And if you see on here, the muscle layers are going to be arranged into two layers. This is the general rule in the gastrointestinal tract. General rule of the arrangement of the muscularis externa or the musculosa, that you do have two muscle layers. We've got exceptions for this general rule. We've got an internal circular muscle layer. The circular muscle layer is located on the inside, and the longitudinal muscle layer, which is going to be located on the outside. This is the general rule. We have exceptions for the general rule, like as we're going to see in the stomach. The stomach is going to have 3 muscle layers, not just 2 muscle layers. We're going to have the pharynx, for example. Pharynx will have the internal muscle layer is going to be the longitudinal muscle layer, and the external muscle layer is going to be the circular muscle layer, which is not like the general rule. Again, the general rule for most of the parts of the gastrointestinal tract, you have an internal circular and an outer longitudinal muscle layer. To the outside of the muscularis externa, we've got the serosa. And remember, the serosa is going to be a layer of connective tissue, a very delicate layer of connective tissue that you're going to have in here. And to the outside of it, we've got a layer of epithelium. This what will be forming the outer covering of the gastrointestinal tract tube. So again, what are the 4 main layers building up any part of the gastrointestinal tract tube? So we can see on here, we've got the mucosa. Underneath the mucosa, we've got the submucosa. Then to the outside of the submucosa, we've got muscularis externa or musculosa, and the outermost layer is going to be my serosa. A 4 stars diagram that we're looking at in here is showing the sagittal section of the oral cavity along with the pharynx. If you see on here, this is a diagram that will be showing information that we've already discussed back with the upper respiratory system. But again, it's really important for us to review this information. Remember, the upper boundary of the oral cavity, this is going to be my palate. And remember, the palate has a bony part, and this is what we call the hard palate. And we've got the muscle part that is located posterior to the hard palate. This is going to be my soft palate. We're going to see an arch here that will be located between the palate and the tongue. We call this arch is my palatoglossal arch. Glosso, something related to the tongue, we call it glossal. So the arch in here that is going to be located between the palate and the tongue on the side of the oral cavities, this is my palatoglossal arch. If you remember, we've seen part of the soft palate that will be pending in the midline at the connection between the oral cavity and the oropharynx. We've called this is the uvula. And remember, the uvula function was to be retracted to close the upper respiratory tract when you are swallowing. So when you are swallowing, you're allowing water, you're allowing food to pass to the oropharynx, which is a part of the pharynx located posterior to the oral cavity. We don't want to have water coming up to the outside from your nose. So when you are swallowing, you're gonna be contracting the uvula, and this will prevent the backflow of the food or fluids that you're gonna be swallowing to pass into your nasal cavity. If we see on here, located in the oropharynx. Here, this is going to be on the side of the oropharynx. We've got the palatine tonsil located inside the oral cavity, representing a major part of it. It's going to be this large muscle. This is the tongue. And remember, in the tongue, we've seen a tonsil, and we call the tonsil here, this is a tonsil of the tongue, and something related to the tongue is going to be either glosso or lingual. So we're going to call the tonsil here that I see in the tongue, it's my lingual tonsil. If we kept moving down, we will see a cartilage that is located above the larynx. This is my epiglottis. We can see also a bone to which many of the muscles that will be moving the tongue and forming the floor of the oral cavity is called the hyoid bone. As we keep moving down, we can see the voice box on here. All this is my larynx, and the part of the pharynx located posterior to the larynx is called my laryngopharynx. So again, the part of the pharynx located posterior to the oral cavity, this is my oropharynx. Here, the part of the pharynx located posterior to the larynx, this is my laryngopharynx. And if you remember, underneath this voice box, gonna have this tube through which the air gonna be conducted down to your lungs. This is gonna be the trachea, and posterior to it, we've got a muscular tube through which the food and liquids gonna be traveling down to the stomach, because this is the esophagus. Any problems so far with this diagram? Any questions? Everything is good so far? All right. Looking here at another diagram. This is going to be a 3 stars diagram. It's much less important compared to the previous ones. It's an interior view to the oral cavity. We can see here, this is going to be the upper lip. This will be the lower lip. And here is going to be the tongue. So identify those 3 structures before you start studying this diagram. You will see that you've got a mucus attachment between the upper lip, lower lip, and the tongue on here. Each of them has a mucous membrane attachment. For the upper lip, it's going to be attached to the mucus covering of the maxillary bone. For the lower lip, it's going to be attached to the mucus covering of the mandible. For the tongue is going to be attached to the floor of the oral cavity. So what do we call those mucous membrane attachments? Those are the frenulae. So you've got frenulum that attaches the upper lip, a frenulum that attaches the tongue, and a frenulum that attaches the lower lip. So what do we call the ones that attaches the superior or the upper lip? Is going to be superior labial frenulum. Labial related to the lips. How about the one that attaches the lower lip to the mucus covering of the mandibles? Is going to be my inferior labial frenulum. And if you remember, something related to the tongue is called either glosso or lingual. So we're going to call the frenulum that attaches the tongue on here to the floor of the mouth. This is going to be my lingual frenulum. Lingual frenulum. If we look at the hard palate, what can we see in the hard palate? The hard palate will have this line that splits it into two halves. This is called the palatine raphe. And posterior to the hard palate, we've got the soft palate. And pending down from the soft palate, in the midline here of the oropharynx and the oral cavity, we've got here, this is my uvula. If you notice on here, we've got two arches. An arch that will appear as a connection between the palate and the tongue. And another arch that appears to be a connection between the palate and the pharynx. So what do we call the one in the front here that appears as an attachment between the palate and the tongue? This is going to be my palato. I'm going to change the palate to the tongue. So something related to the tongue is glossal or lingual. So we're going to call this arch is my palatoglossal arch. And the one that appears to connect the palate to the pharynx on here, located posteriorly, this is going to be my palato, and related to the pharynx is going to be pharyngeal arch. So two arches that we can see on here, one located anterior to the other. The anterior one seems to be connecting the palate to the tongue is called the palatoglossal arch, and the posterior one that appears to be connecting the palate to the pharynx is going to be my palatopharyngeal arch. Located in between, we've got the tonsils on here. And those are the tonsils that will get inflamed if you've got an infection when you are a child. We go to the pediatrician, he will ask you to protrude your tongue and say, what he's looking at inflammation in those tonsils on here, on the sides of your oropharynx. We call those tonsils that might get inflamed, and if they got so large or they are exposed to recurrent infections, we're going to need to perform a tonsillectomy. We're going to remove them. We call those are the palatine tonsils. Those are my palatine tonsils. So when you are studying the diagram, start again with upper, lower lip and the tongue. Then move on to the frenulae, superior labial frenulum, inferior labial frenulum, and the lingual frenulum. Then look at the palate. Check the hard palate, check the palatine raphe, then check the soft palate and the uvula. Then look at the arches. We can see an arch that seems to be connecting the palate to the tongue. This is going to be my palatoglossal arch. And another one that seems to be connecting the palate to the pharynx. This is my palatopharyngeal arch. Located in between, we've got the palatine tonsils. If we're looking here at the lingual frenulum, we can see those tiny openings on both sides. So we've got large ones on here. Those are the openings of a large gland located underneath the floor of the mouth. This is going to be the submandibular gland. So those are the openings of the submandibular ducts. And we've got multiple small ones on the sides away from the midline. Those are the openings of smaller glands, smaller salivary glands, as we're going to see in the next diagram. Those are the opening of the sublingual duct. All right. Yeah, so if a person has a problem with the attachment of the tongue, here, it's going to be something very rare and it's going to be a congenital anomaly. Yes. It's not going to be present on its own. So this person is going to be born with other defects as well. Will the class last until 2 p.m.? I hope to finish earlier, but most likely yes. Most likely yes. All right, so if you remember, chemical digestion is initiated in which part of the gastrointestinal tract? Where do you first start the chemical digestion? This is something that we discussed in our last meeting, in the oral cavity, in the mouth, exactly, in the mouth. A much less important diagram is going to be a 2 stars diagram showing the tongue. What you can see on here, we have the papillae on the dorsal surface of the tongue. We've got 4 different types of papillae, and those are the only thing I want you to know for this diagram. Those to be able to identify those 4 papillae. So if we're looking here, we can see the most widely distributed type of papillae that we have on the tongue. This is going to be the filiform papillae. Those are the whitish papillae that are covering most of the surface of the dorsum of the tongue. Scattered in between, we can see those reddish dots. Those are the fungiform papillae. In the posterior part of the tongue, we can see on the sides, on here, we've got the foliate papillae and arranged in a V-shaped line in the posterior part of the tongue. We've got the circumvallate papillae. All those papillae, except for the filiform papillae, will act as taste buds. So each one of them gonna be able to detect specific chemicals, and they will be responsible to detect the taste. The only exception here is going to be the filiform papillae, which are going to be given the roughness to the dorsal surface of the tongue, which allows you to mix the food while you are chewing. All right, so again, this is a two stars diagram. The only the only thing that you need to be able to identify on this diagram again is going to be the different types of lingual papillae. Again, the most widely distributed all over the dorsum of the tongue, those whitish papillae everywhere. Those are again the filiform papillae. Those reddish dots scattered between the filiform papillae, those are the fungiform papillae, appearing as a V shaped line in the dorsum in the posterior part of the tongue. Those are my circumvallate papillae, and occupying the posterior one third of the lateral parts of my tongue, those gonna be my foliate papillae. It's the same. It is the same. Circumvallate or vallate. It is the same. A three stars diagram. The only things that we need to identify here, or mainly, is going to be the salivary glands. Remember, we've got pairs three pairs of sets of salivary glands. We've got the parotid gland that will be covering the angle of the mandible. And this here is going to be the parotid duct through which the parotid gland going to be releasing its secretions. Located below the tongue, we've got the sublingual salivary gland. Remember, the openings are located away from the lingual frenulum. This is my lingual frenulum on here. And located surrounding the mylohyoid muscle, we can see here another salivary gland. This is the submandibular gland. And remember, the submandibular gland gonna have larger ducts that will open just on both sides of the lingual frenulum, on the lingual frenulum. So important structures again that we need to identify on this diagram will be the parotid gland, sublingual gland, submandibular gland, of course the tongue, lingual frenulum, and parotid duct. So again, important structures would be one, parotid gland, parotid duct, submandibular, sublingual, and the lingual frenulum. Those are the five most important structures that you need to identify on this diagram. All right, this is again a three stars diagram. Moving on to the anatomical structure of the stomach. The diagram showing the anatomical structure of the stomach is going to be a 5 stars diagram. It's a highly important diagram. If we see on here, there's going to be the muscular tubes that will be conducting the food down from the pharynx towards the stomach. This is called my esophagus. It travels through the diaphragm. It opens up at this region in the stomach. This is called the cardia. And the cardia here will have a sphincter that regulates the passage of the food down from the esophagus to the stomach. So we're going to call the sphincter in this region, it's the cardiac sphincter. This is the cardia of the stomach. We can see a dome-shaped part of the stomach. This dome-shaped part of the stomach is going to be the fundus of the stomach. Fundus. The mid region on here, all this part is going to be the body of the stomach. All this region, all this mid region of the stomach is going to be the body of the stomach. The most peripheral part of the stomach, this is going to be the pylorus. If you see on here, the pylorus has an entrance. We call this the pyloric antrum. And I have a constricted region. We call this the pyloric canal. And I have a muscle that regulates the passage of the food from my stomach to the proximal part of the small intestines. This is called the duodenum. All right, so we're looking here at again the different parts of the stomach. This is one of the most important diagrams for the digestive system. We're looking here, this is going to be the esophagus. The regions that will be surrounding the entrance of the esophagus into the stomach, this is called the cardia or the cardiac region. The dome-shaped part of the stomach, this is the fundus. The mid region here, this is going to be the body of the stomach. The most distal part, this is going to be the pylorus. Remember, we've got the pyloric antrum. The constricted part on here, where I have the most vigorous contractions in the stomach taking place. This is called the pyloric canal. And I have the sphincter on here. that will be regulating the passage of the food. We don't call it food anymore. After it gets partially digested in the stomach, we call it the chyme. So the chyme is going to be passing here from the stomach to the proximal part of the small intestine. This is going to be my duodenum. So what did we study? Did have a fundus, a body, and a terminal region. Do you remember? What body organ did we study that had a fundus, body, and a distal region? And the distal region also did have an internal opening and an external opening and the canal in between. Did we have uh, fundus in the urinary bladder? No, no. Yes, Mary. Yes, the uterus. Remember? The uterus. The stomach reminds me of the uterus. Why? If you remember, the uterus did have a dome-shaped structure, dome-shaped part. This is called the fundus. It has a mid-region. This was the body of the uterus. And the terminal regions that passes in the vagina, we call this is the cervix. And remember, this terminal region, the cervix, it did have internal opening, external opening, and the canal. It did have internal os, external os, and a cervical canal. So here's the stomach. Here's the stomach. We have a fundus, we have a body, and we have the pylorus that corresponds to the cervix on here. If you look, it has an entrance, it has the pyloric antrum, it has a canal, pyloric canal, and it has an opening to the outside is surrounded by the pyloric sphincter. All right, does this make sense? Does this make sense? Also, if you see on here, the stomach gonna have a large curvature, This is gonna be my greater curvature. And this much smaller one on the other side, this is gonna be my lesser curvature. If you notice on here, the muscle layer of the stomach. Remember what was the general rule? We've got an internal circular muscle layer and an external longitudinal muscle layer. Here in the stomach, we have 3 muscle layers rather than 2. We've got 3 muscle layers. We've got longitudinal muscle layer to the outside. We've got circular muscle layer to the inside, but to the inside of the circular muscle layer, we've got an extra layer. This is my oblique muscle layer. Oblique muscle layer. Why do I have 3 layers in the stomach? Compared to my esophagus, it's gonna have 2 layers. Compared to my small intestine, compared to my large intestine, it's gonna have only 2 layers. Why is the stomach gonna have three layers compared to the rest of the gastrointestinal tract? It's gonna only have two muscle layers simply because the stomach is a bag, not a tube. If you're talking about the esophagus, the small intestine, the large intestine, we are looking at tubes. Here in the stomach, I have a third dimension that I need to contract in, which won't be satisfied by constrictors and the longitudinal muscle layer. I need a third dimension to contract within, which is the oblique direction. So again, this is to remind you that the stomach wall has three muscle layers, not only two muscle layers. You're going to have an outer longitudinal, middle circular, and inner oblique muscle layer. Again, why do I have this extra oblique muscle layer? Simply because this of the structure of the stomach. The stomach is like a bag. It has a third direction that it needs to contract within. Unlike the rest of the gastrointestinal tract, those are tubes. So they will either constrict or perform a peristaltic movement, which are both satisfied by the circular muscle layer and the longitudinal muscle layer. But I need here to contract in an angle to perform this contraction to be able to move the food around in this bag. I'm not a tube, I am a bag here. All right, so again, this is a very important diagram, a 5 stars diagram. I would say even 10 stars, it must be on your test. So please answer this diagram as your in-class activity for today. So I will be drawing the different parts and please put them please identify those different parts of the stomach and the structures related to the stomach. We got this region on here. right, so please identify the different structures. Again, let me answer this with you. So number one, what did you put as number one? Any answers here? This is my esophagus. And number two, what did you put for two? This is gonna be the cardia, exactly. And the dome-shaped part of the stomach, this is my fundus, exactly. The main core of the stomach, this is gonna be exactly the body. And remember, this terminal part, the most distal part of the stomach, this is gonna be the pylorus. And the entrance of the pylorus, this part on here, this region, is called the pyloric antrum. And then we have this constricted region in the pylorus. You remember, what do we call this? This is my pyloric canal. And what do we call the muscles that will be guarding the exit of the chyme from the stomach to the small intestine? We call this the pyloric sphincter, exactly. And what do we call the proximal regions? This C-shaped part of the small intestines is going to be my duodenum or duodenum. And what do we call this large curve of the stomach? This is my greater curvature. And what do we call the smaller one? Lesser curvature, exactly. If we're looking closer to the structure of the wall of the stomach, we're going to see that the mucosa, it looks different than the mucosa that we've seen at the beginning of the chapter here. So the mucosa that we've seen in this general structure of the gastrointestinal tract tube, it was flattened. Here it appears to have pits. So I have pits on here. But it's still the same structure. I have epithelium, I have connective tissue underneath. This epithelium, this is my lamina propria. I have a thin muscle layer, muscularis mucosae. The major difference that you will see is gonna be not in the submucosa. Submucosa is gonna still the same. The major difference is gonna be the presence of a third muscle layer in the muscularis externa. So if we're looking on here, we've got outer longitudinal muscle layer, we've got middle circular, and the most internal muscle layer is going to be the oblique muscle layer. This is going to be the two major differences that we have. You have the mucosal lining is going to be arranged in pits, as you see on here. The other main difference that we can see in this diagram compared to this general diagrams that we've seen is going to be the presence of a muscle layer, an extra muscle layer, which is the internal oblique muscle layer. All right, this diagram is a three stars diagram. Three stars diagram. Moving on to the small intestine. The small intestine is going to be formed of three main parts. We have the most proximal part, and it is the shortest of the three segments. It's my duodenum. And it's going to be the C-shaped part that will be surrounding the head of the pancreas and will be receiving the food from, or the chyme from, the stomach. Located distal to the duodenum, we've got the jejunum, which is the middle segment of the small intestine, and it's going to be about 8 feet in length. Distal to the jejunum, we've got the ileum, and it's about 12 feet in length. And it's going to be carrying the remaining part of the food that remained unabsorbed and undigested to be conducted to the large intestine. The part of the large intestine that receives this undigested, unabsorbed part of the food was the cecum, this pouch-like structure that's going to be receiving the undigested, unabsorbed part of the food, the remaining part of the food that the digestive system failed to break down and absorb. It's going to be this cecum. So the ileum is going to be conducting the remaining part of the food to the cecum, and we call the junction here between the ileum and the cecum is going to be the ileocecal valve. I have a valve that allows a unidirectional movement. Here you don't want to have feces moving back from the large intestine to your small intestine. You want only to allow a unidirectional movement here from the small intestine, from the ileum to the cecum, and not going backwards from the cecum to the ileum. So again, three parts of the small intestine that we've seen. First one is my duodenum, the shortest region. Next is going to be the jejunum, see on here, which is 8 feet again in length. And the most distal part, and actually the longest part of the small intestine, is going to be the ileum. Why is the duodenum is so important? Because this is where you will be receiving the secretions from the liver and from the pancreas. Also, you're going to be receiving the food from your stomach, receiving the chyme, the partially digested food from the stomach to the small intestine. This is how the duodenum gonna look like. A 10 stars diagram is gonna be the diagram showing the relationship between the duodenum liver, pancreas. It's really, really important. And it's kind of confusing a little, so please pay special attention here with me for us to break down this diagram. What can we see on here? We can see on the side the liver. The liver is formed of 4 lobes. We have right lobe, left lobe, caudate and quadrate lobes. But what we can see from this view is just the right lobe and the left lobe. This is the right, this is the left lobe. One of the major functions of the liver, if you remember, was to form the bile. And remember, we did mention a little about the bile when we studied the life cycle of the red blood cells. If you remember, red blood cells, when they get old, after they remain in the bloodstream for 100 to 120 days, they will be broken down and the hemoglobin gets released to the outside of those old cells that will be destroyed, and the hemoglobin will get broken down into heme and globin. And remember, the globin part is a protein part that gets reused, broken down into amino acids. You use it to build up other proteins, and you've got uh, the heme part, in which you're going to be recycling the iron, you're going to be transporting the iron on the transferrin, and the porphyrin ring will be conjugated and will form the bilirubin that will be conjugated and released as part of my bile. So the bile that we're going to be releasing here contains bile salts, the bilirubin. It's going to be collected from the liver cells through the ducts of the liver into 2 large ducts. We call those are my right and left hepatic ducts. So from the right lobe, we've got the right hepatic duct. From the left lobe, we've got the left hepatic duct. They both will fuse together. So they will together form what we call the common hepatic duct. The common hepatic duct gonna be fusing with the duct coming from the gallbladder, which is a muscular sac located on the ventral surface of the liver. This muscular sac located on the ventral surface of the liver on here, it's my gallbladder. And the duct of the gallbladder is what we call the cystic duct. So common hepatic duct is carrying now the bile coming from right and left hepatic ducts. This bile is about a liter per day. So you are releasing large amounts of bile per day. So you don't want to get rid of all this amount of bile directly to the small intestine. You would rather get it stored and concentrated until you get food reaching your small intestine. So most of the bile here that will be conducted down is going to be conducted down from the common hepatic duct will be directed towards your gallbladder. For the gallbladder to absorb the water out and concentrate the bile until you get food into your stomach that is about to get released in the proximal part of the small intestine. The C-shaped part of the small intestine again is my duodenum or duodenum. All right. So as I get a signal, which is a hormone that gets released from the gut, it's called cholecystokinin or CCK. This cholecystokinin is going to be stimulating the gallbladder to contract, telling the gallbladder, yeah, we've got food arriving to the small intestine, to the duodenum. So the gallbladder contracts and is going to be squeezing the biles, which has been concentrated out through the cystic duct and then to the duct that will be connecting both cystic duct and the common hepatic duct together. This is my common bile duct. All right, so many ducts in here, so let's repeat them. So you've got here right hepatic duct, left hepatic duct. They fuse together, they will form my common hepatic duct. Gallbladder has a duct. What do we call this duct of the gallbladder? This is my cystic duct. Cystic duct and the common hepatic duct will fuse together and they are forming a common duct through which the bile going to be traveling, either that has been concentrated or hasn't been concentrated yet. We call this duct is my common bile duct. All right, this is my common bile duct. Any problem? Any problem so far? Anybody is still here? All right. So we're looking here at the pancreas. The pancreas has three main parts forming its structure. We have the head of the pancreas, the body of the pancreas, and the tail of the pancreas. Head, body, and tail. Those are the three regions here forming the pancreas. So another name for the common bile duct is some textbooks would say just bile duct. All right, so some textbooks will call it just bile duct rather than common bile duct. All right, so again, the structure of the pancreas on here, what we're looking at is this going to be the head of the pancreas, body of the pancreas, and the distal part on here is just going to be the tail of the pancreas. As you see, the duodenum is going to be surrounding the head of the pancreas. The pancreas is going to be responsible to release a major part of the digestive enzymes that will be responsible to break down the foods that we're eating, perform a large part of the chemical digestion. So those enzymes and the pancreatic juice that we are releasing from the pancreatic cells going to be released through small ducts into a larger duct that is traveling throughout the length of the pancreas like this. This is going to be my main pancreatic duct. The main pancreatic duct, as you see on here, will be meeting with the bile duct, and they will form together a bulging connection before they join together the duodenum. So the bile is coming here from the common bile duct or the bile duct. The pancreatic secretions are coming here through the main pancreatic duct, but you see they will be passing through a common pathway, which is this bulging part. We call this region on here, this enlarged duct that will be receiving secretions from the liver and from the pancreas is called my hepatopancreatic ampulla. Hepatopancreatic ampulla, and the hepatopancreatic ampulla are going to have a muscle surrounding it regulating the passage of the secretions. So we're going to call this is my hepatopancreatic sphincter. So what do we see on here? We see this enlarged part, enlarged tubes that receives secretions from both the main pancreatic duct and the common bile duct. We call this is my hepatopancreatic ampulla. Why did we call it hepatopancreatic ampulla? Because remember, I'm receiving secretions from the liver, from the pancreas. I am an enlarged duct. Remember, where else did we see an ampulla? Where else did we see an ampulla? Recently, in the previous chapter. Male, yeah, I've seen it in the male, but it wasn't that prominent. The ampulla of the vas deferens. Is this what you're saying? Yes, we've seen it in the male reproductive system, the ampulla of the vas deferens, but we didn't stress on it in the male reproductive system. We stressed on it in the female reproductive system. If you remember, the ampulla of the fallopian tube is the site, normal site of the fertilization. Remember what was the structure of the fallopian tube or uterine tube? We did have those finger-like projections that will be picking up the ovulated egg. What do we call those finger-like projections again? Fimbriae. And we've got the entrance here of the tube. We've called this is my infundibulum. And then we've got this bulging region. This is the normal site of the fertilization of the eggs. This is my ampulla. And remember the junction between the uterine tube and the endometrium. This was the isthmus. Right? Do you remember those structures? So again, what makes the ampulla of the uterine tubes so important? This is where the fertilization normally takes place. This is where the fusion between between the sperm and the egg will be taking place. All right. All right, so this is the most important. If I would say this, if I'm picking only one diagram, but this is not real. You're gonna have many diagrams from the digestive system. If I would be picking a single diagram to be testing you on in the digestive system, is gonna be this diagram showing the different parts of the bile ducts, as well as the relation between the liver, pancreas, and the duodenum. All right, so let's do this as another in-class activity. Right, so please name the different structures of this diagram. So let me double check here at what number did we stop last time. So we stopped at 11. Any questions? Any questions? Anything which is not clear on this diagram? All right, so we can have a break for 10 now, and we come back, discuss one remaining diagram, one remaining important diagrams that we still have in the digestive system, and we gonna have our introduction of the metabolism chapter. All right, so let's have a 10 minutes break. We come back, discuss more about the digestion and metabolism. Number 23 is this bulging part that's gonna be resulting from the fusion of the blue and yellow. Is this clear, Happy? All right, welcome back. Let's have a look on here, this diagram, before we start our last diagram of the digestive system. Can you hear me or not? Yes. All right, great. So we're looking here at this diagram. Remember what we can see on here. This is going to be the liver. This is going to be the right lobe. This is going to be the left lobe. For number 12, the duct that will be allowing the bile to travel from the cells of the left lobe. This is going to be my left hepatic duct. The duct that will be collecting the bile from the liver cells from the right lobes is going to be the right hepatic duct. And the bile that will be collected from both right and left hepatic ducts going to be traveling into a duct here, a common duct. We call this is my common hepatic duct. Remember, this muscular sac that will be located on the ventral surface of the liver, this is called the gallbladder. And the duct here, through which you're going to be squeezing out the concentrated bile, you don't have it on the diagram to be marked. This is my cystic duct. The duct that gets formed from the fusion between the common hepatic duct and the cystic duct. This is my bile duct or my common bile duct. Any of them is going to be a description of the duct, bile duct or common bile duct. Bile duct or common bile duct on here is going to be meeting with a duct coming from the pancreas, collecting the pancreatic juice from the pancreatic cells. We call this is the main pancreatic duct. Remember, the main pancreatic duct along with the bile duct will form a bulging duct. We call this the ampulla. And the ampulla that receives the liver secretions and the pancreatic secretions, we call it the hepatopancreatic ampulla. And remember, it's going to be surrounded by a muscle that allows the regulation of the passage of those secretions to the small intestine. So it has a sphincter. So we call this the hepatopancreatic ampulla and its sphincter. Remember, the secretions here from the pancreas and the liver are going to be released to the proximal part of the small intestines. This is going to be my duodenum. The pancreas, if you remember, it has three main parts: head, body, and tail of the pancreas. Any questions? Any questions? Anything which is not clear in this diagram? All right, great. So another five stars diagrams that we're looking at today is going to be the diagram showing the different parts of the large intestine. So in the small intestine, we have been completing the mechanical and the chemical digestion. So we did complete all the digestion possible. We did allow the absorption. So when you review those recordings, those video recordings I will be posting, we are discussing in those recordings how the absorption of the different nutrients is going to be taking place. So after I complete the absorption of all the nutrients, the remaining part of the food that we ingested that failed to get absorbed and or digested, it's gonna be remaining in the terminal part of the small intestine. If you remember, small intestine was formed of three parts: the duodenum, the jejunum, and the terminal part was the ileum. The ileum is the longest part of the small intestine. It's going to be conducting the remaining part of the food down to a pouch-like structure. This is called the cecum. And the cecum has a protruding part like this. This is called the vermiform appendix. The vermiform appendix doesn't play a major role neither in the digestion or defecation. Some studies do suggest that it's important to re-establish the normal bacteria flora in case of a prolonged intake of antibiotics. So in your large intestine, you have normal bacteria living inside. So if you are using a lot of antibiotics, those bacteria are gonna be destroyed. And this will give a chance to pathogenic bacteria to take their place. So studies did show that the vermiform appendix will not be affected by the intake of those antibiotics because the antibiotics will have less access to it. So it serves as a backup store for the good bacteria that lives in your large intestine. The undigested and absorbed part of the food will be conducted from the cecum upwards. This part of the large intestine is what we call the ascending colon. It's going to be traveling up, carrying the undigested and absorbed part of the food upwards in the colon. Then the colon flexes and it's going to be traveling in a horizontal pattern. So we call this part of the colon that travels horizontally. It's the transverse colon. This flexure that we have is what we call the hepatic flexure. Why do we call it the hepatic flexure? Because simply it's going to be on the same side as the liver. So you've got the liver on the right side of your abdominal cavity. So the flexure of the colon on the same side as the liver, related to the liver, is going to be the hepatic flexure. Transverse colon carrying the remaining part of the undigested and absorbed part of the food will be traveling down. We call this part of the colon that travels down, this is my descending colon. And the descending colon will be conducting the feces now into an S shape, a flexed part of the colon. We call this as the sigmoid colon. Then sigmoid colon will be conducting the feces to a straight part of the large intestine. We call this as the rectum. If you remember recta is straight, so the straight part of the large intestine is called the rectum. And finally, we're going to be passing through a short canal that is guarded by sphincters. We've got internal and external sphincters guarding the anal canal. And the opening of the anal canal to the outside is going to be the anus, through which the feces is going to be traveling to the outside. If you see on here, the flexures that I have between the transverse colon and the descending colon is going to be on the same side as the spleen. So on the left side, we've got the spleen. On the right side, we've got the liver. So the right flexure is called the hepatic flexure also. And the left flexure is called the splenic flexure because it's on the same side as the spleen. Hepatic flexure is the same as right colic, yes. For Ophelia, the right colic flexure is another name for the hepatic flexure, and the left colic flexure is another name for the splenic flexure. All right. If you will see here, you're going to have the attachment, the membrane attaching the transverse colon. We call this the transverse mesocolon. Transverse mesocolon. All right. Much less important structures for us would be the epiploic appendages. Those are protrusions to the outside of the colon. And you see on here, this is going to be my tinea coli. Those are much less important structures for us. Much less important structures for us. The tinea coli and the epiploic appendages. The constrictions on here that we see in the colon, in the throughout its length. Those bulging eminences here, those are the host ray, and single one is going to be the rostrum. A very important structure that we've mentioned earlier is going to be the valve that will be located between the ileum and the cecum, which allows a unidirectional movement of the feces from the ileum to the cecum and prevent its backward movement. So the valve located between the ileum and the cecum is my iliocecal valve. So again, what are the important structures that we need to identify on this diagram? Most importantly would be ileum, cecum, ileocecal valve, the vermiform appendix, ascending colon, right colic flexure or the hepatic flexure, transverse colon, splenic flexure or left colic flexure, descending colon, sigmoid, rectum, anal canal, and anus. Less important would be the transverse mesocolon, the rostrum, and the least important for us would be the epiploic appendages and the tinea coli. All right, again, the diagram showing the different parts of the large intestine is going to be of high importance. Again, all the 13 structures on here on this diagram are going to be the most important to be identified. Comes next would be the transverse mesocolon and the rostrum. Least important to be identified are going to be the epiploic appendages and the tinea coli. It's just a manifestation of the appearance. This is due to the constriction. It doesn't like the rostrum is like a description, like the gyri, for example, of the brain. Those bulging, those elevations on the cerebrum. It's a structural unit. All right. Questions? Let me do it different way here. Go to this. Oh, it won't. So let me reduce the shared screen for you to see better. I believe this gonna. All right. Is this good? Is this good? All right, so those are the most important diagrams that we have in this chapter. Again, we've got four main diagrams. I'm sorry. We've got six main diagrams from the reproductive system, those ones that we've mentioned last class. And we've got here in the digestive system, those are the main diagrams. This is first one. This will be the second one, third, fourth, diagram number 5. One of the most important, this is number 6. It shows the different parts of the stomach. 7, the wall of the stomach is much less important. Must be on your test is going to be the diagram showing the duodenum and the relationship to the liver and the pancreas, along with the ducts carrying the bile and the pancreatic secretions. Very important diagram that shows the structure of the large intestine. So I will double check. I will double check it's not if it's not shown on Canvas. I believe I did upload it before, but let me double check. All right, so I will be posting a similar diagram, a similar presentation that will contain all those diagrams. No problem. Optional final exam. If somebody is looking to uh, attend the optional final exam, I will collect the registration over this week. So I will, for those ones who are gonna be, just stay tuned, check the announcement whenever I start to collect the names for those who are willing to make the optional final exam. All right, the optional final exam is going to be a substitution for a missed lab exam or a lab exam that you want to make up, like you didn't do good on. All right, this is for the optional final exam. For the lecture exams, please email me if you did miss any of the previous lecture exams, and I will find a way for me to give you a substitution for the uh, the lecture exam. Yes, you so you are required to complete 4 lecture exams and 4 lab exams. All the 8 exams are required. The only optional exam is going to be the final exam. Yes, so last lab exam is going to be available for you by the end of the day today. It's going to be available for you by the end of the day today. Right, so it's going to be accessed as what you did with the uh, as what you did with the third lab exam, the blood and respiratory lab exam. Uh, it's going to be available again through smarter proctoring. You enter through smarter proctoring. You should now have figured out how to attend a proctored exam. So some people are asking me about if I can reduce the file size on the exam for the page to load faster. So some people did have a problem with loading the questions on uh, on the blood and respiratory system. The problem here that I have is if I did reduce the file size the, of the diagrams, I'm afraid that it won't appear clearly for you on the test. So just be patient. I did reduce it a little, but if you don't have a fast internet connection, and if you are mainly connected through a Wi-Fi network, and your device is located away from the router, you will experience some extra time loading the page. All right, because I use a, like a higher resolution diagrams for everything to be clear on the test. All right, I did receive 2 emails stating that the page is loading slowly. Is this the case for everybody? All right, so for those who did experience some slowing down was uh, the page loading while you are taking the test, this means that your internet connection needs to be assessed. So either you connect your device with a LAN cable, so connect your router directly to your device through a wire rather than a Wi-Fi, and try to load the page, load everything not on a Wi-Fi. Yes, so in order to take the test, you will need to share the screen. You will need to allow the software to access your webcam and microphone. If you at any point mention that you don't want them to access your microphone, you're simply gonna lose your attempt. So you need to be uh, so cautious when you are replying to the pop-up messages that will appear to you. Because simply if you denied them to access uh, your webcam or microphone, this is the whole purpose of the thing. You need to close everything. Nothing should be open while you're taking the test. This is the test. You can't open anything else other than your exam. And you need to not have multiple screens attached. You need only to have one screen. You are monitored. you are monitored. You need only to access the exam while you're taking the exam. You can't enter the password. So I got lost. So I got lost. So Mary, you've had technical difficulty, but were you able to complete the test or not? All right, so let's have a conversation at 2 o'clock. All right, so for those who did experience some difficulty, some technical difficulty, can you stay and help other people who had similar difficulty? I will open the microphone for you to share your experience. So I would be grateful if you can do this. All right, I will stay with you after 2 o'clock. So for you to experience, to tell us your experience with the difficulties that you had, and please, if somebody still having a difficulty with the test, please remain after we're done. After at 2 o'clock we're gonna be done, and you can remain after 2 o'clock. So for us to discuss how can we resolve those issues. All right. The exam is still open until midnight tonight. All right, but you need to complete the test before midnight tonight. So you need at least to start maximum by 11 after you figure out everything related to the smarter proctor. Yes, for the final exam is going to be proctored. Yes, final exam is going to be proctored. Yes, the optional final exam is going to be proctored. This is not the final optional, the optional final exam. This is our last lab exam that includes the reproduction and the digestion. All right, the final exam is optional, yes, and it's going to be proctored. The topic of the final is going to be everything. It's going to be cardiovascular, it's going to be blood and respiratory, it's going to be the four labs combined into one exam. All right. Questions. All right, so in this chapter, we are discussing the metabolic reactions. If you remember what's metabolism by definition, metabolism is simply all the chemical reactions which are taking place in your cells, all chemical reactions, all chemical reactions. We classify those chemical reactions according to whether I am breaking down or building up. If I start with a simple chemical compound like glucose, and I'm storing this glucose into a more complex structure like the glycogen. So I'm building complex compounds from simple ones. So we call this is an anabolic activity or anabolism. What if I'm breaking down a protein, which is formed of amino acids attached to one another, into their simplest form, which are the amino acids? So I am transforming a complex chemical structure into a simple chemical structure. So we call this as a catabolic activity or catabolism. So again, all the metabolic activities gonna be either falling under catabolism or anabolism. If I'm building up, it's an anabolic reaction, and this is gonna be part of the anabolism. If I am breaking down, it's a catabolic reaction, and it's gonna be part of the catabolism. Why would I be breaking down? In most of the cases, to get the structures that you can deal with to build up your own dishes or to generate energy in the form of ATP, adenosine triphosphate. So how the energy is stored in an ATP? ATP stands for adenosine triphosphate. So it has 3 phosphate groups attached to the adenosine. The attachment here between the adenosine and this phosphate group, it's a high energy bond. This is where I have the energy. So if I am building up this bond, I will lose energy. If I break down this bond, I will be releasing energy. So this is a stored form of energy. If you have energy, you transform the energy into a bond between adenosine and the phosphate. If you want to get energy, what you're gonna do is you break down the bond and the energy gonna be released. So what I'm trying to do here is transform high energy bonds stored in the glucose, which is C6H12O6, for example, into high energy bonds stored in an ATP that I can use. I can't directly use the energy by breaking down this glucose. I need to transform those high energy bonds from the glucose to build up high energy bonds between an ADP, an adenosine diphosphate, and adenosine attached to 2-phosphate, and one other phosphate This is the bond that I will be building up by breaking down the high energy bonds located in this glucose. What is the single most chemical reaction that takes place in your body? Is the oxidation of glucose. Glucose is C6H12O6, has 6 carbons. What slide? Like this one, the black one here, you see here. You see this one. You can see the carbohydrate metabolism. I just fixed it. I didn't do anything. All right, no problem. All right, so now you're good. So you can see both on here. All right, great. So, single most important chemical reactions that takes place in your body is again the oxidation of glucose. I break down the glucose, C6H12O6, in the presence of oxygen. And this is going to result in the formation of carbon dioxide, water, and I will be producing ATPs. So I need 6 oxygens. I need 6 oxygen to break down a single glucose molecule. And this is going to result in the production of 6 carbon dioxide, 6 water, and around 32 ATPs. In other words, you did transform 32 ADP. You attached each one of them to another inorganic phosphate. This results in the formation of 32 ATP. How are you saying 32 and here on the slide it says 36? So we're gonna see that not all the energy that's gonna be produced from the glucose oxidation is gonna be transformed into ATP. There will be some of it that will be transformed into heat. And that's why it's not gonna be an accurate number. So it's not gonna be definitely 32 or 38, it's gonna be 32 to 36 to 38. It's gonna be a range. It's according to what? According to the amount of energy that will be lost as heat. Not all the energy that I will be producing here is going to be transformed into ATP. Some of it gets lost as heat. So it's a range between 32 to 36 to 38 ATPs. You're going to see even some textbooks would say 38 ATPs, all right? So I started with glucose, and remember, this glucose is going to be circulating in my blood, and in order for it to enter my cell, this is my cell membrane, it needs to have a transporter, glucose transporter or GLUT. In order for my cell to form those transporters for the glucose, they need to receive a signal that tells them, yes, we've got glucose outside, please form the transporters to carry this glucose in. So the signal here is going to be the insulin that's going to be produced from the pancreas, it's gonna bind to the surface receptors on my cells, and this will induce the cell to produce the glucose transporters that will be allowing the glucose to pass to the inside of the cell. So can I break down the glucose if I don't have insulin? No, because you simply won't move this glucose to the inside of the cell. If you didn't have insulin or you have defective insulin receptors, there will be no influx of the glucose to the cytoplasm. And so this metabolic reaction will never take place because simply you don't have glucose inside your cell. In the cytoplasm of the cell, in the cytosol, this glucose is going to be broken down into two molecules. Those are my pyruvic acids. Two pyruvate or pyruvic acids. Here, each one of them is going to be containing 3 carbons. So remember, glucose has 6 carbons. Here, if you split the glucose into 2 halves, this is going to be 3 carbons each, pyruvate or pyruvic acid. This reaction is what we call the glycolysis. Glycolysis is taking place in the cytoplasm. And this is going to yield 2 net ATPs. 2 ATPs that will be produced just by breaking down the glucose into 2 pyruvic acids. How many ATPs should I be producing from each glucose again? Around 32. So I did split the glucose without oxygen in the cytoplasm of the cell into 2 pyruvic acids. Each of the pyruvate, if I did have enough supply of oxygen to the cell, this pyruvate gonna be traveling to the inside of the mitochondria, and on its way, it's gonna be transformed into an acetyl-CoA. If we're looking at the first step of the glycolysis, I didn't only split the glucose into 2 pyruvic acids, I have lost also hydrogen. In order for this hydrogen to be taken off the glucose, it needs to be carried on a hydrogen transporter. And the hydrogen transporter here is going to be NAD. So now you are getting rid of 2 hydrogens. So I have 2 NADH that will be formed. And I've lost energy. I'm producing 2 ATP. The transformation of the pyruvic acid into acetyl-CoA is going to be allowing you to get rid of one carbon dioxide, of one of the carbons in the form of a carbon dioxide. And in the same time, you're going to also be losing a hydrogen. This hydrogen again is going to be carried on a hydrogen transporter, an NAD, so now it's going to become an NADH. What happens next? The acetyl-CoA gonna be passing through a cascade of chemical reactions on the inside of the inner membrane of the mitochondria. If you remember how the mitochondria did look like, it had an outer membrane and an inner membrane like this. So here we've transported the acetyl-CoA to the inside of the inner membrane of the mitochondria. Acetyl-CoA, in order to pass through this cascade of reactions, it's going to be binding to oxaloacetate. This is a 4-carbon structure. Now, after I've lost this carbon dioxide, what do you think? How many carbons do I have in the acetyl-CoA? So each pyruvate, each pyruvic acid had 3 carbons. If I've lost a carbon dioxide from each of them to become an acetyl-CoA, this means the acetyl-CoA has how many carbons? Anybody still here? All right. So we're looking here at glucose. Let me repeat everything again. Glucose, what is the structure of the glucose? What is the structure of glucose again? Structure of glucose, chemical structure of the glucose. C6H12O6. What this means? I am formed of 6 carbons, 12 hydrogens, and 6 oxygen. This glucose, in order to pass through the inside of the cell, I need to have a glucose transporter, or GLUT. In order for me to have a GLUT, the cell needs to receive a signal from outside called insulin. And the insulin needs to bind to its receptor. This is what stimulates the cell to produce the glucose transport. So now I allowed the glucose in the cell. So now I have glucose C6H12O6 in the cytoplasm of the cell. The very first step in order for you to break down the glucose by oxidation is to break it down into 2 pyruvic acids. So each of them has how many carbons? If the glucose is formed of 6 carbons, when you split it into 2 exact molecules, each of them gonna have 3 carbons. We call this step is the glycolysis. Glucose lysis breakdown. During the glycolysis, I'm not only breaking down the glucose, I'm also losing hydrogen. And in order for me to lose hydrogen, this hydrogen needs to be carried on a hydrogen carrier. So the carrier here is called NAD. Each of them gonna carry a hydrogen. I am losing 2 hydrogens in this reaction. So what is the total number of hydrogens that a pyruvate will have? What do you think? If I've lost 2 hydrogens. So again, my question. I did have a glucose, and a glucose has how many hydrogens? 12. You split it into 2. And when you split it into 2, you've lost 2 hydrogen. So each of the two gonna have how many? So 12 minus 2 divided by 2 is gonna be 5. Make sense? Make sense here? All right. So what we know, this pyruvate has 3 carbons, and each of them has 3 carbons, and each of them gonna have 5 hydrogens. And during this reaction, don't want to get into the details of it, we're gonna have a net gain of 2 ATP. So I will be breaking down 2 high energy bonds in the glucose, and I will be using those high energy bonds to build up 2 other high energy bonds between 2 ADP and 2 inorganic phosphates. This results in the formation of 2 ATP. Actually, it's not that easy. We have many reactions that will be taking place in here just to transform a glucose to 2-pyruvate. This is a very simplified reaction. Actually, we have multiple reactions on here. But what we need to know is that from this reaction, you're gonna end up having 2 ATP, net gain of 2 ATPs. Anything seems confusing so far? All right. In order for me to complete the breakdown of this glucose, now that became 2-pyruvate. I need to carry the pyruvate to the inside of the mitochondria. If you remember, what was the mitochondria responsible for? It was the one responsible for the production of energy. And here we are studying how the mitochondria are going to be producing the energy. So the mitochondria, if you remember, is formed of 2 membranes. Look in here. The very first step is going to be to transform the pyruvate into another chemical structure, passing to the inside of the inner membrane of the mitochondria. The pyruvate will need to be transformed into acetyl-CoA. During this reaction, what's going to happen? You're going to be losing one of the carbons, producing 1 carbon dioxide. So how many carbons are still going to be remaining in the acetyl-CoA? If each of the pyruvates going to be losing a carbon dioxide. So you're going to have only 2 carbons remaining in the acetyl-CoA. And you're going to be losing 1 hydrogen from each of the pyruvate carried on an NAD that became an NADH when it carries the hydrogen. So how many hydrogens are still remaining in this acetyl-CoA? Remember, each pyruvate did have how many? It did have 5. So if I'm telling you that you're going to be losing a hydrogen, transforming each of those pyruvates into an acetyl-CoA, this means that you're going to have only 4 hydrogen remaining per acetyl-CoA. Makes sense so far? All right. Inside the inner membrane of the mitochondria, we're going to have enzymes that will carry on a cascade of chemical reactions that allows the breakdown of the acetyl-CoA. In order for the acetyl-CoA to pass through those cascade of reactions, the first step is to bind to oxaloacetic acid. The oxaloacetic acid, when it binds to the acetyl-CoA, it's going to be forming here citric acid or citrate. This citric acid, as it passes through the cascade of reactions, it's going to return to become oxaloacetic acid. So what did I lose throughout those reactions that I didn't mention here, which are on this diagram? See, I've lost hydrogen, I've lost carbon dioxide. What did I lose throughout those reactions I did pass through, starting from citric acid to become oxaloacetate? So again, what is the citric acid is formed of? Citric acid here is formed of oxaloacetate and acetyl-CoA, right? Where did you get the acetyl-CoA from? You've got the acetyl-CoA from the pyruvate, which is coming from the glucose. So this is the glucose part on here. So what is the citric acid is formed of again? It's formed of acetyl-CoA and the oxaloacetate. If I told you that this citric acid is going to pass through a cascade of reactions on here, it will become oxaloacetate. So what did I lose throughout those chemical reactions? The acetyl-CoA, right? Anybody didn't get this idea? So citric acid is what? Is both acetyl-CoA and oxaloacetate. When you are passing through the chemical reactions, the citric acid, you see on here, will become oxaloacetate another time. So what am I releasing here are parts of the acetyl-CoA. All right, so again, citric acid is acetyl-CoA and oxaloacetate. If I told you that the citric acid becomes oxaloacetate, this means which part is lost? You see on here, the acetyl-CoA. Is this clear? Clear? We're going to be building up on this information in a second. All right. So how many hydrogens do you expect to lose from each of the acetyl-CoA passing throughout those reactions? We call those reactions is the Krebs cycle. Actually, the guy who did first discover the Krebs cycle got a Nobel Prize. Or another name for the Krebs cycle that we are currently more commonly using is the citric acid cycle. All right, so what do you expect to lose through each Krebs cycle? What do you think? How many carbons do you expect to lose? How many carbons do you expect to release in the form of carbon dioxide from each Krebs cycle? 2. Remember, you've got 2 carbons in each of the acetyl-CoA, so you're going to be losing 2 carbons. So you're going to be releasing 2 carbon dioxide. And how many hydrogens do you expect to lose through the Krebs cycle? How many hydrogens? Remember, acetyl-CoA through this specific part of the Krebs cycle. Through this specific part of the Krebs cycle. Remember, how many hydrogens did we start with? 4, right? So we're going to be releasing 4 from this cascade of reactions. 3 going to be carried on NAD. This will become NADH. And 1 will be carried on a different type of hydrogen carrier called FAD. So it becomes FADH. So what is the total number of hydrogens that I've lost in the Krebs cycle, including the transformation of the pyruvate into acetyl-CoA? Seeing 1 in here, 1, 2, and 3. So you've got the 5 hydrogens that we started with from each pyruvate. How many Krebs cycles are you going to be carrying on for each glucose? What do you think? How many Krebs cycles would I have for each glucose? Remember, each glucose is producing 2 pyruvic acids, and each one of them going to be transformed into acetyl-CoAs that will pass through this cascade of reactions. So how many hydrogens did I lose through the Krebs cycle from the glucose? I have lost 10 hydrogens. Five hydrogens from each of the pyruvic acids. Each of the pyruvic acids gonna be losing 4 hydrogens attached to NAD that will become NADH, and 1 that will become an FADH. This takes place in both pyruvic acids. So I have a net gain of how many NADHs from each Krebs cycle. I've got 8 NADHs, and I've got 2 FADHs. Again, this is not from each pyruvic acid. This is from each glucose. So each glucose, started here again, did pass through glycolysis. So it did release 2 NADH. And as the pyruvate passes into the Krebs cycle, each of the two pyruvates gonna be yielding 5 hydrogens, 4 of them attached to NAD, and 1 of them is gonna be attached to an FAD. Those are the 10 remaining hydrogens in the glucose molecule. Remember, glucose has how many hydrogens? It has 12 hydrogens. Those are the 12 hydrogens that it did have. We've seen 2 released in the glycolysis attached to NAD, and 10 released in the Krebs cycle, 8 of them attached to NAD, and 2 of them attached to FAD. Also, in the Krebs cycle, you're gonna be breaking down high energy bonds that will be directly forming ATPs. You're gonna be producing 1 ATP for each of the Krebs cycles. So this means I am producing a total of 2 ATPs from the Krebs cycle per glucose. Anybody still didn't get confused? Anybody didn't get confused? All right, so let me repeat this again one more time. So I started with glucose C6H12O6. I split the glucose into 2 pyruvic acids. So each of them gonna have 5 hydrogens. Why? Because I've lost 2 in this reaction, and I've formed 2 ATPs from the first step, which is the glycolysis. From the glycolysis, I've released 2 ATPs, and I've lost 2 hydrogens carried on to NAD. So I've got 2 NADH. I did transform each of the pyruvic acid, each of the pyruvate into acetyl-CoA. So the acetyl-CoA inside of my inner membrane of the mitochondria are gonna be passing through a cascade of chemical reactions called the Krebs cycle. The transformation of the pyruvate into acetyl-CoA and the Krebs cycle reactions, all this gonna be yielding 8 NADHs, 2 FADHs, and 2 ATP from each glucose. Why? Because simply you're gonna have 2 Krebs cycles per each glucose, as you do have 2 pyruvate. So how many ATPs did you get so far? How many ATPs? You've got 2 from the Krebs cycle, and you've got 2 here from the glycolysis. So far, I did produce 4 ATPs. That's it. How many did I tell you at the beginning that you're gonna be producing? Around 32. So where are the rest of the 32? This will take us to our last step in the oxidation here, which gonna be the electron transport chain. So here we're looking at the 2 membranes of the mitochondria. This is my outer membrane of the mitochondria. This is my inner membrane of the mitochondria. Inside of the inner membrane, what do I have? I have NAD attached to hydrogen. This is a high energy bond. High energy bond. The hydrogen carrier is going to be binding to the hydrogen. Same for another hydrogen carrier. Here is going to be my FAD. Those are located where? Again, in the inside of the inner membrane of the mitochondria. On the inside of the inner membrane of the mitochondria, you've got surface carriers on here in the inner membrane of the mitochondria that will be acting as enzyme to split this bond between the NAD and the attached hydrogen, and others to split the bond between the FAD and the hydrogen to which it's attached. I split them apart and I will allow the attached hydrogen to travel between the two membranes of the mitochondria. So here I was located on the inside of the inner membrane. The hydrogen now is going to be accumulated in the intermembrane space. Again, this is a very simplified way to look at this step. This is not an accurate information. If you're taking biochemistry, you're going to discuss this in details. All right, this is a very, very basic and simplified, inaccurate information. Just to give you an impression about what's going on. As you accumulate lots of those protons in the intermembrane space, this creates a very high potential energy. So this high potential energy is going to be used while the hydrogen protons or the protons are going to be trying to travel from their high potential energy between the two membranes to the inside of the inner membrane of the mitochondria. I will allow to pass down their pressure gradient, passing through a transporter here. This is called my ATP synthase. And as the hydrogen is traveling, it's going to be losing its potential energy. But you know, energy can be lost or created. Energy gets transformed. So the ATP synthase, what is it doing? It's transforming the potential energy of the hydrogen traveling down from the intermembrane space to the inside of the inner membrane of the mitochondria into a chemical bond between an adenosine diphosphate and an inorganic phosphate. I'm creating this high energy bond using the potential energy of the hydrogen that travels in. And this allows me to create an ATP from those hydrogen carriers. So what if I kept allowing those hydrogen to travel inside, those protons to travel inside of the inner membrane of the mitochondria? They will reach equilibrium. They will be balanced across the membrane and no further hydrogen will be able to travel inside. That's why those hydrogen will be binding to oxygen, and that's why you're breathing. You're breathing to supply your cells with this oxygen that will bind to this hydrogen coming into the inside of the inner membrane of the mitochondria for you to build up water. So each 4 hydrogens that you did have, and how many 4 hydrogens did I have in my glucose, remember, it's C6H12 and O6. So how many hydrogens? 12 hydrogens. How many 4 hydrogens did you have? Anybody still here with me? 3. So you need 3 oxygen. For you to build up the water. So, but remember, you can't have single oxygen. So you're gonna need to have on here for the carbon dioxide to be formed in earlier stages, you need also oxygen to get rid of the carbon. Remember this. All right, so again, why am I breathing again? For me to keep the hydrogen at much lower pressure inside of the inner membrane of the mitochondria to keep allowing the hydrogen to travel from its high potential energy in the intermembrane space to its lower potential energy passing through the ATP synthase. All right, so this completes our discussion for today. Let me answer this. For the metabolism lab handout, it's a self-explanatory assignment. So it will tell you to count the calories that you're taking per day. The key here is to allow an equal intake to the output. So it will give you the equations on how to calculate your body mass index for you to check whether you have obesity or you have your body mass index is lower than normal or higher than normal, you are overweight or underweight. Those are the equations that you're going to be using. It's a self-explanatory. You're going to be just calculating your basal metabolic rate according to your body size using the equations provided. One thing only that you will need to know here is going to be it will ask you the inspiratory reserve volume, expiratory reserve volume. So if it's asking you about a lung volume or capacity, consider yourself normal. So use a normal value back from the respiratory chapter. Use a normal value for any lung volume or capacity it will be asking you about. So it will tell you what is your lung volume or capacity. So check out the normal values back from the respiratory chapter. All right, yes, 6,000 is going to be the total lung capacity. That's here we're going to be using different ones. So it will ask you, according to the lab, the respiratory lab, what was your respiratory volume, let's say, or inspiratory capacity. So we didn't measure the lungs volume for ourselves. So consider it as a normal value, and you can get the normal values from the respiratory system chapter. All right. Any questions? Any questions? So this is an interesting assignment. You can just go over it It's a self-explanatory. You're gonna submit this as a separate assignment, not with the in-class activities, not with the rest of the in-class activities. You're gonna be submitting this under Unit 4. I will be modifying, so it wasn't requiring a submission. I will modify this to require a file submission. So you're gonna be uploading the file once you get it completed. Everything is going to be in a single file. Everything is going to be in a single file that you're going to be submitting here. If you go to Unit 4, in-class activities and pop quiz, you will see here, you submit all the in-class activities, including the lab work, all combined into a single PDF file that you will be able to upload on campus. We won't have for the metabolism, you're gonna be uploading this where it says metabolism lab handout assignment. So you have two files that you're gonna be uploading. In-class activities and pop quizzes. This includes all your in-class work, including the labs, into a single file. The other files that you're going to be uploading is going to be the metabolism lab handout. Yeah, you can only write down the answers, the parts that you need to fill in a separate Word document, and you can upload it. Or if you would like to print them out and then scan all of them with completed pages, this is going to be also fine. So either or. So submit it the way you want. In-class activities are worth 160 points from your grade. In-class activities are not going to be extra credit. No. This is all the works that we have been doing throughout the semester. Labs, in-class activities, pop quizzes that we've completed in class and on the virtual meeting. How is this going to be graded? It's going to be curved according to you. So your grade is going to be determined according to your class performance. Yes, no problem. Just complete the ones that have been assigned to you. Just complete the ones that have been assigned to you. So we didn't have an in-class activity with every single class. I know this. Any questions? Any questions concerning the structure, the modifications on here? Again, for the metabolism lab handout, you're gonna be submitting this as a separate file directly to the metabolism lab handout under Unit 4. And for the rest of the in-class activities that you have completed, it's going to be submitted separately, all together as a single file in here in the in-class activities and pub quizzes. All right, so if I told you how many assignments you did have throughout the semester, I would be ruining the idea of it. The idea is that A person has been attending all the classes, completing the assignments as they are given in class. This is a marker of your attendance. If I came at the end, I told you those are the assignments that you should have been completing throughout the semester, this simply means I'm telling you to not attend the whole semester and come get the work at the end in the last week. This is gonna not be fair for the people who have been attending. Is this clear? So please don't ask me how many assignments should I have been completed, should I have completed. You should be aware of them. Last lecture exam 4 is also going to be available for you to complete starting from tonight. So you have one lecture exam, one lab exam still remaining to complete. Lab exam is the one that will be proctored. The lecture exam is not going to be proctored. This will be our last lecture exam. Yes, this will be our last lecture exam. So it should be available already for you. You see on here, lecture exam 4, it actually has been posted and one of you did already complete this. So lecture exam 4 is available for you to complete until April 28th. All right, for those who have problems with smarter proctoring, please remain connected. We're gonna have a 10 minutes break and